All right, next up we've got B23. Section B23 is all about cell specialization. I told you, if you watched the other video, I told you we'd come back to this one. And so we have cells, and they start unspecialized. So all of your cells right after fertilization are unspecialized, or also known as undifferentiated. So they're just chilling. They're not doing anything yet. And so you have basically the zygote is the first cell, the fertilized egg. And so that will then split into like a blob of cells, which form the early embryo. And as it forms the early embryo, you get more and more cells, right? That should have been an even number, right? Because I'm going to divide at the same time, but that's fine. So it continues to go like this. And then what happens is you have your like matrix of the cells. They're secreting these chemicals. They can kind of tell what they are. You create these different proteins. And as these proteins are created, they're created in different concentrations. And so the concentration of the protein will affect the gene expression of the cells. And so gene expression is regulated by the different chemicals that are created, the different proteins. And so gene expression, remember, is going to be turning on or off. And so this leads to cell specialization. So cell specialization is going to be regulated by turning these genes on or off. And so they'll start to differentiate into the cells that they're supposed to be based off of the cell environment and the proteins that are created. Well, why can they do that? Well, what we have is stem cells. And so stem cells are what we start with. And so stem cells are cells, the definition are cells to divide endlessly and differentiate along different pathways. Most of your cells are obviously not stem cells, which is why, right, you can't just magically restore function and you can't regrow limbs or anything because you don't have those kind of stem cells. And so what we've got going on, right, is that's our definition. The two characteristics, right? The two characteristics are ability to divide endlessly and that they can differentiate. So they're currently undifferentiated, but they can undergo differentiation to become different types of cells. And so where do you have adult stem cells? I guess we'll talk about that first. So let's talk about adult stem cells. There's lots of different types of adult stem cells. The ones we're going to focus on for this class are bone marrow. These are the ones that become our blood cells. And skin stem cells. And so even skin stem cells can become a couple different things, right? They can become skin cells, our traditional skin cells. They can also become the ones that produce sebum, so they're like oil secreting ones, or the ones that can grow hair. There's kind of three different types they can become. So they can only become like those three things, give or take, but they're much more limited. So speaking of limited, what are our types? So there are totipotent, which is early stage in the embryo. It can become anything, including the placenta. Right, so extra embryonic tissue. Pluripotent can become anything in the embryo. And then multipotent, which can become multiple things, but not a lot, right? Multiple, and this is typically found in adults. Okay? So those are the three main types of stem cells. Oh, and I wrote 3, but this is standard number 4. I forgot to write 3 up there. Sorry, that's standard 4. My bad. All right, number 5. One of the things of specialization is cell size. And so the different sizes are a way of specializing the cells. And so you have your sperm is really tiny because you need to make a lot of them. Whereas our egg cells are huge because you only release like one every once in a while, and by once every 28 days, give or take, right? So it's got to be huge. It's got to have a lot of nutrients because it's going to become the zygote. So it's huge to have all those nutrients in it. Red and white blood cells. So red blood cells are going to be a little smaller, and so are white blood cells. They're both going to be small, red and white blood cells. Why are they small? Well, they have to go into capillaries. So these really narrow passageways. So they're very small to fit in the narrow passageways. Unlike our neurons, which are huge, some of your neurons can be up to 1 meter in length, okay? 1 meter in length. So the one that runs from basically your spinal cord all the way down to the tip of your foot can be about 1 meter in length. So neurons are huge, and your striated muscle fibers are going to be much larger as well. So your striated muscle fibers, are going to run through your muscles, your skeletal muscle, and so they're going to be much longer as well to help with contractions. They're nice and long for contraction. Great. All right. Why are cells different sizes, though? Why are there limits? Why don't they just grow endlessly? Why aren't we one giant blob? Well, we learned a little bit already, right? Multicellularity helps with cell specialization. But also, there's the surface area to volume ratio. Surface area to volume ratio is one of the main driving factors. So surface area is all about your ability to transport in and out. Your volume tells you how much you need and how much waste you produce. So it's those things. So it's how much energy you need to bring in versus how much you need, how much can you get in versus how much you need, how much you can get out versus how much you produce. And so the surface area to volume ratio is a ratio, so it means it looks like this. Surface area is a squared function of size. Volume is a cubic function. So if you increase the size of a cell, this one will increase, but this one will increase faster. So the ratio will go down. So if you increase cell size, the ratio goes down. Of course, you can have adaptations to make it so that doesn't happen, but this is the overall principle, right? So if you increase the cell size, the ratio goes down, which means the cell is less likely to survive. It's going to either have too much waste that it can't get rid of, it can't bring in enough energy, whatever it might be. And so it's messing up this ratio. So surface area is all about transporting things in and out, and volume is all about the amount of energy you need, like energy and nutrients you need, and the waste you produce. And that is the end of the S cell stuff. Moving on to the H cell sections, standard 7. So we said you can have adaptations to increase the surface area to volume ratio. One of the ways you can do it is you can flatten the cell. That increases the ratio. Flatten the cell, brilliant, way to go. This is an example of this. Ooh, look at this. Standard 8 is a type 1 pneumocyte. Is a flat cell for gas exchange. So it's super thin, like ridiculously thin. I spelled thin wrong. Super thin. Okay. Flattening the cell. What else can you do? You can also do microvilli. So microvilli are finger-like projections. on the outside of cells, and so they look like this. So in your convoluted tubule, right, you can have these projections out. These will increase absorption because they're increasing surface area. And you can also have invagination. No, that's not this. And those are like inward folds. So these are like outward folds. And these are like inward folds. This is invagination. Inward folds. Oh, that spelling is hard. Another example of a flat cell is like our like red blood cells. They're pretty flat. So an example for this one is our erythrocytes. Wow, that's a big word. I hate it when they do that. Erythrocytes are our red blood cells. They're the same thing. They're just being obnoxious. And then this one is found in the proximal convoluted tubule of the kidney. Proximal convoluted tubule of the kidney. And so this is going to increase absorption in the kidney. We'll learn more about the kidney later. We learned about type 1 pneumocytes. They have that surface for gas exchange. And then type 2 pneumocytes, because if there's a type 1, there must be a type 2. Type 2 pneumocytes are adapted for secretion. So how are they adapted for secretion? Well, they have the microvilli to increase secretion, and they also have lots, they have a huge like ER, right? Lots of ER, because that's what's going to create the package to be excreted. Okay, standard 9. Cardiac muscle cells. Versus striated muscle. So cardiac muscle cells have these gap junctions that allow for electrical impulses to be transmitted across them. Because they stimulate themselves, they also have more mitochondria because they use more aerobic respiration. Whereas our these are singular cells, so these are like normal cells. Whereas these are like a bunch of cells grouped together with no junctions between them, right? So in these ones, they're striated because they are like one cell that became a whole bunch. So the fiber, is it really a cell? Well, kind of, but it's really a bunch of cells with no partition. And so this allows it to act as a single unit with multiple nuclei. So that is our cardiac muscle and our striated muscle. The last one we already talked about, right, is our sperm and our egg. So the sperm has a tail, and it has lots of mitochondria because it's got to move that tail. Whereas the egg has a protective layer, and it's really large, so it has lots of nutrients in it. I think those are kind of the main things. We'll talk about them more later. Yeah. And that is B23.